we just try to highlight that what way this particular material is there. I will just first, before proceeding to any lichen, I will just highlight one particular our work for this Lemania. Lemania, it is a like red algae. We have seen a green algae, blue-green algae, and also we tried to see, because this Lemania moniporensis, it is found in northeast India, we could see how this red algae, whether they are very efficient or not. So we took a HPLC grade water and we prepared algal extract of this Lemania. The Lemania moniporensis, it is our new report from the world, because Lemania moniporensis, the particular species with molecular entity, it is not reported earlier. So we named them in the state as a Manipur state as moniporensis. It's our foreign collaboration of this particular authentication, and we could see that this particular alga, how efficient of this particular manganese or oxide nanoparticles. So we prepared extract and we could synthesize manganese oxide nanomaterials, and you can see that yes, when potassium permanganate solution is being seen, and it is forming a colloidal manganese nanoparticles. We try to authenticate with our UV visible, and again, and thus, with the absorption spectrum, and we could see that this particular nanomaterial absorption peak of 520 nanometer, indicating a complete reduction of manganese, and it is being formed. And the band gap, the band gap energy, it could be determined that how this particular equation it is being found at particular volt. So we could see that yes, nanometal it's being formed, and it is giving some different bands parameters of the peak. The peak is coming in a particular band, assigning the different materials it is there, and we could see the time analysis and highlighting a very interesting type of a so much the side pattern distribution, and the particularly HRTM images showing a lattice fringes, and the particle size distribution it is showing a as a synthesized particle is obtained measuring the size particle 35 particles from the corresponding TM image. So what I mean to see here, it is indicating a polycrystalline nature, and it has a specific spacing of how much different corresponding interplanar separation, and this is some same picture of that, which is showing the agglomerated small cluster of this particular manganese nanoparticles, and it is showing several macro particles also, while some individual particles are also clearly visible. So what I mean to say here, this phytomolecules which are among the which are present, the hydrogen bonding between that and the surface of the nanoparticles might have caused the aggregation, because when the algae is being given, which has a huge type of a type of a poly material, the maybe of some particular metabolites is off significantly, acting on that, and particular phytomolecule molecules which have a specific hydrogen bonding is making a type of aggregation and helping and the nanomaterials to be formed. So we have also characterized the phytomolecules of this particular alga. I'm not going to highlight all those things. So this alga, whether it is showing some of the rhodamine B, the degradation dye. Rhodamine B, particularly, it is one of the important dye, and we could try to see that is because it is a manganese nanoparticles. We try to apply this particular degradation of rhodamine B, and as well as the methylene orange and methylene blue. So here we could see that this particular in the three conditions 1, 2, A, B, C, and we could see that the rate of degradation was found to be not very much time for it is taking of 6 into 10 to the power 2 minutes, and the rate of degradation, particularly the rhodamine B, is significantly higher and the as compared to other, and maybe lowest in case of methylene blue. So it depends on that why it is that because of this manganese nanoparticles, it is facilitating very faster rate of degradation for this rhodamine B dye. Okay, so and this degradation efficiency is also very high, as I just told that, and that we have plotted it, and this is giving us a how much again. So if it is degrading fast, it is okay, but how much efficient? It is showing us 86%, 0.8% for this rhodamine B, followed by 72.1% for methylene orange, and for methylene blue it's 67. But okay, it is another thing that again we applied for a energy means it is not an energy intensive method. We simply followed this laboratory scattered sunlight condition. We are not artificially applied any light source to facilitate the degradation of this particular dye. So this is one of the most important aspect of our work, and effect of this catalyst, particularly uh, photocatalyst dose on dye degradation was also examined, that which particular percentage of this particular catalyst is working. So poor degradation of the dye molecule was observed for the degradation without manganese nanoparticle, and degradation efficiency increases as a function of concentration of the photocatalysis dose. We are identifying that in the scattered. Scattered means it's a in the laboratory condition diffused light. No artificial light has been added. So whenever we are increasing the dose of this manganese nanoparticles, we are getting a good photodegradation efficiencies. So the photodegradation efficiency appear to be strongly influenced by the number of active sites, the photon absorption ability of the catalysis, because it is getting some energy, and we are we have compared again. Let's see that what this red algae is, how much efficient it is there, and we tried to see that yes, the time is taking of this it's okay, and particularly this algae is also well efficient of the efficiency, the degradation efficiency, particularly this particular. So, of course, I am not telling that yes, we can achieve 100% or even, but it is comparable. It is comparable because 86%, it is all approaching towards other a well-known biological entity which have a degradation efficiency of this rhodamine blue. So this algae is nevertheless is a very far, rather it is very close to this particular other well-known, well-authenticated organisms. So this has also been published in Good Impact Factor Journal in Organic Chemistry Communications, and now this yes, this is another alga. It is a marine alga, that is Ulva profunda, and we could get it from some marine belt, and we prepared the algal powder, and we could see that is nickel oxide nanoparticles we have produced, and we have seen how efficient this nickel oxide nanoparticles with precaution. So I will move little fast, maybe I have to complete before my time. And the UV study, it is also showing, yes, it is indicating at a particular absorption is prominent in the visible region particularly. And the peak is at 315 meter, and it is showing a good formation of that. FTI studies highlighting the nickel nanoparticles is at particular band and giving us the peak of different assigning of different things. And TM analysis of this particular nickel oxide, it is also showing the side picture, it is showing the polycrystalline nature of the, when you talk about the poly, that means there are a formation of a different like band or different corresponding to the different plane, and these nanomaterials are being encircled with a proper spacing of this that, so different size it is being available, and we can see very nicely it has been formed on that. Okay, so we could see again scanning picture, it is showing a highly agglomerated like nanomaterials, it is there, and it is being formed. I'm not going to much detail of that. So this nanomaterial again this nickel oxide nanoparticles, where we have examined of this particular nitrophenol, 4-nitrophenol. I would have examined that it has been reduced to 4-aminophenol using a very laboratory scattered sunlight. I'm always highlighting the light source. We have not used any direct sunlight. We have not got performed the experiment in the outside, that is exposed to the sunlight to get the energy source. No, and we have also not in the laboratory. We have not also not applied any artificial light source. So it is absolutely not energy intensive method. So it is absolutely eco-friendly way of formation and then photocatalytic reduction. So the graph is absolutely showing within after 27 minutes, you will like we can see that after 27 minutes, once we apply this particular reduction starts, then it is giving us a very good result and confirming the kinetic pattern, and the efficiency, it is also showing very nice, very fast. Okay, and 86% of the efficiency it is finally showing in the like time. And photocatalytic reduction, it is synthesizing of this nickel oxide nanomaterial. It is for the reduction of the nitroaniline by using a receiving electrons from BH4 ions and transmitting them to the aromatic nitro compounds. So this whatever it is there, it is being the electron is being shared, and it is being used, and it is being allowing to reduce. So that is one of the most important aspect of that. Again, after 36 minutes, it is showing the photocatalytic degradation, uh, and the degradation efficiency is 92% of the efficiency of that particular aspect we are. So every minute, almost every 3 minutes, we have proceeded, and we could see 96% of the degradation efficiency. It is being shown this nitroaniline dye. Okay, and there are some of this aspect of this antibacterial activity of these nanoparticles, and it is also showing us some good result. Uh, Maybe it is being applied, and we can this marine alga ulva we have published in inorganic chemistry communication, and like same this alga. I'm so sorry. This alga again we have applied for this is the final aspect of my work, and the tin oxide we have seen, yes, this ulva whether how efficient this can be there. So we have again applied some of the tin chloride and used that, and again seen that yes, some particles has been formed at a particular 2 to 7 nanometer. The absorption peak is being there, confirming the FTIR aspect of different bands, peaks, and again TM analysis. We could see that the side pattern, and the so interestingly, the particle size is greater than 5 nanometer, and there is a very interesting polycrystalline nature, and the different line in different line, it is corresponding to the different plane. the ring it is being there. So it is exhibiting a very interesting concentric rings, and that has been of this facile silver phase it is being showing. And we could see that yes, there are some of this SEM analysis of synthesized tin quantum dots are there. What we see that emergence of some micro structures with special structural features, very typical type of a cuboidal type of a rod, and the broken edge indicating the ultra small particles. They are tightly packed in the assembly. So the presence of a distinctive edge point of this plausible application of this tin material for catalysis. So they may be this particular the edges. The edges is facilitating the different catalysis, chemical sensing, and also biological sensing. So let's see what application of this particular material can be there. Maybe again we could see that photocatalytic degradation of this methylene blue, like methyl orange, and in the laboratory again in the scattered sunlight as light source, okay, and the laboratory condition. And the rate of the degradation blue is very fast as compared to methylene orange. So methylene blue rate of degradation is very fast and as compared to this, and we could see the efficiency, and we could get that yes, 70 minutes and maybe 35 minutes methylene blue, and methylene orange we could proceed because the efficiency was indicating little higher in the more time thing. So at 70 minutes we could see that yes, most of this the efficiency it is the maximum. Then what were great application of this tin nanomaterials, methylene blue photocatalytic degradation. Very interestingly, we could see that we could the rhodamine blue. This we could again compare with this methylene blue and try to see. Yes, this is showing a very interesting degradation mixture, degradation like mixture of that simultaneously. Even if we put it a mixture of our nanomaterial to this particular blue, means it is, see, as we why we did it, because we know that in the nature no dye or no pollutant, it is been exposed singly. So there is always a mixture. So we could see that yes, let's see that mixture of this particular to dye, what is there, and which particular degradation of the which particular blue rate of degradation is high. So methylene blue rate degradation is high as compared to the rhodamine B. Rhodamine B. So we could try to work on that, and efficiency we could see simultaneously. We have this is very interesting work we could publish, and we could highlight about this thing. So there are also some metal sulfide nanomaterials investigation of this photo physical, catalytic, and biological activity. So again, some of these cadmium sulfide nanoparticles using a blue-green alga, we could see that yes, synthesis has been done of this cadmium sulfide. We could confirm it with from 41 to 410 to 475 nanometer doing the FTIR analysis, and we could do some XRD a pattern, prominent peaks we could see it corresponding to the different analysis, the conformation, and the widening peak imply a small particle size of the product. So the synthesized cadmium sulfide nanomaterials, we could see the again the spacing, and it is corresponding to the 111 plane, and the average particle size is so small, 3.87 nanometer. So possible reason of the appearance of the aggregates could be due to the large concentration of the sample loaded on the grid, as well as the duration of the probe sonication applied to disperse of the powdered sample into solution. So it may be that reason that why a aggregate has been formed. We could get a nice aggregate from here. But again, we did some of the same analysis, and The SEM is absolutely highlighting the closed, packed, fine, agglomerated, interconnected spherical nanostructure, and the agglomeration is maybe by the hydrogen bonding, as presumably it is maybe of OH and NH among the phytomolecules on the surface of this nanometal. So why I'm highlighting about the phytomolecules? Why there is a bonding between, I'm highlighting because they are the main ways which facilitate this particular activity of these nanomaterials. So this cadmium sulfide nanomaterials, yes, what a great activity of this particular is doing for rhodamine B. It is being shown with the extent of this catalytic degradation. It is easily monitored by decreasing the UV visible peak at 460 nanometer, as well as the change in the color also. So it is also very evident, and pseudo first order kinetics it is showing, and for methyl orange also it is showing, but yes, it is showing is a different pattern. The extent it can be monitored by the decrease of this 460, and it is showing us a good information. So methyl orange and rhodamine B degradation efficiency, if it can be compared, then of course it's absolutely close, little higher at the methyl orange, and then as compared to rhodamine B, but nevertheless it is showing around the 90% efficiency at the 80 minutes exposure. So this is another cadmium sulfide nanoparticle activity. It is showing this particular alga, and this we have published the Nostogarnium and in this Bio Science. And again, for the chloral ellipsoidea, which is the first algae, we could see the copper sulfide synthesis, and we could confirm with that and with FTIR analysis. And the TM picture is so interesting. It is showing some of this particular presence of this corresponding to the copper and sulfur. So which is a doubly agreement of this, and it is showing a very interesting type of agglomerated lump, which is reveal the number of aggregates. The number of aggregates, it's very important to understand that how much the looseness or the aggregation it is there. So the homogeneity of this the composition, it is the good degree of dispersion it is highlighting. But anyway, I just try to highlight about the application part of this methylene blue of this particular algae. Yes, it is showing as a good peak at particular the 662 nanometer, and the copper sulfide photocatalysis by degradation efficiency. It is also showing the degradation was observed a little poor without copper sulfide nanoparticle. With copper sulfide nanoparticle, it is showing a good result, and we could we could publish it in a inorganic chemistry communications. And this again zinc sulfide nanoparticles of this vulva profunda, we could analyze, synthesize, and analyze. And the photoluminescence study we could see, and the FTIR result, it is authenticating for a nanomaterial formation, and the particles is also the polydispersed and were composed of many well dispersed nanoparticles in clusters. So what type of clusters or type of the agglomeration it is there, it also shows that how efficiently it can degrade or it can be applied for this particular nanomaterial. It has also showing some of the good antimicrobial activity, and preparation of the Cyphus spirophorides lichen extract we have prepared, and we can see that this particular alga, this is one of the final thing I will highlight about the RGO, the graphene oxide. As we all know, this graphene oxide, it is one of the most important entity of this nanomaterial. Nowadays, it's being applied, and we have with silver manganese nanoparticle and the RGO silver manganese nano hybrid, we have made a combination of that. And we have found that this particular by UV visible study, this RGO, silver manganese oxide nanomaterial, and we have found that we have compared with the silver, and we see that this is a RGO layer also. It is very nicely, it is being formed. So whenever a sheet of graphene oxide layer is being formed, it is the presence of a electrostatic attraction between the functional group. So this is a ultra thin structure of this particular which is observed in the graphene oxide. So we have compared with the graphene oxide and the RGO which is being there. And again, finally, yes, I am going to complete almost rhodamine blue and methylene blue, whether this has a very well application of this particular graphene oxide. So we have seen also this antibacterial activity and the surfaces and interfaces. We have published this RGO nano hybrid. Due to the paucity of the time, I am not going to highlight, but this is the last part of my talk of that graphene oxide, reduced graphene oxide. Whenever a graphene oxide is there, it has been a very packed. You can see the graphite. You can see the graphite. Yes, graphite. Whenever there is a oxidation or exfoliation, then this is being converted into, because when the oxidation, then we find it, we find it that this particular oxygen OH, it is being entered inside and it make a type of a space. And upon reduction, this you see upon reduction the oxygen has gone, and there is a chemical thermal and electrochemical reactions, and that has been when graphene oxide has been reduced, it is giving us a graphene. And that graphene, it is a chemical vapor deposition, and it is showing a very thin sheet that is reduced graphene oxide. So we have worked on this property. I'm not going to much detail of that. We have prepared this Ulva profunda, this particular application, and we have seen this green synthesis approach of this particular alga, and graphene oxide has been formed, and the composite materials. This is all our work. We have prepared the graphene oxide, and along with the silver manganese, so many metallic, bimetallic nanohybrids we have prepared, and it is showing us a very good result. And this is, out of all the work of our nanomaterials, this is one of the best work which I can highlight. It is degrading the Congo dye, red dye so fast. And yes, I am going to say, and it has been published in Metals Today Sustainability. So this is a green synthesis of like a ternary nanohybrids using a lichen. And as we all know, this graphene for this Russian scientist, Nobel Prize 2010, the Andrew James and the Konstantin the Novoselov, they have got the thing. And using the lichen powder, we have used this particular graphene, reduced graphene composite, and we have applied this particular work for this extraction. And we have seen the photocatalytic, antimicrobial, anticoagulative properties of these nanoparticles. And we have got it for different algae and different lichens. This is the conclusion part. So this type of green synthesized nanomaterials have potential to act as an anti-cancer, antifungal, drug delivery, energy storage agent. So in future, we can extend our research in this domain. And I finally acknowledge to the Department of Microbiology, Assam University, Silchar, for some of our collaboration, and Department of Chemistry, Assam University, BITS Pilani, Goa, and the SAIF Northeastern University, Shillong, for our collaborative effort. Thank you. All things in nature, there is something of the marvelous, and we should recognize them. Happy Women's Day to all of you, and let's be daring and seize every opportunity with steadfast determination and dedication to excellence. Let us celebrate successes, recognize everyone's contributions, and reaffirm our commitment to promoting gender equality and empowering Amen. Thank you all.
Oh, thank you so much, ma'am, for your extraordinary talk. Almost covered all the elements in the periodic table. So thank you so much for the very informative talk. Due to the time constraint, we can allow only one questions from any participant. Any question? We can unshare, then I can.
Yes.
They are do appreciating the talk, very informative session and all, but no question yet.
Fine. And I understand, but because this is the work, I could move it very fast, but it takes little time.
Yeah.
And but anyway, my email ID is jayashriraut8@gmail.com. You all are. I'm happy to collaborate with anyone who are interested, and we can work together to make all this work again more appear like we can always make it more good contribution. Then it is also a pleasure for me to be with you all, and thank you so much. Thank you all.
Thank you so much, ma'am.
Yeah.
Okay. Let's have the final talk of the forenoon session. Let's invite Dr. T.S. Anita from.
I will leave here, Dr. Mubarak.
Yeah, yes, ma'am. You may.
Thank you so
ma'am.
much. Okay. Yeah.
Dr. T.S. Anita from Department of Biotechnology, Pondicherry University.
Shikha, Shivani, Shruti.
Excuse me. I request the participants to please mute and video also locked. Yeah. So Anita is from Pondicherry University, and her area of research is about cancer biology and therapeutics and the repurposing of drugs, and which includes animal biotechnology, ocular biologies and all. She completed her master's and PhD in animal biotechnology from Animal Sciences, Badass University, Tiruchirappalli, and also works as a scientist from CIDRF on Sri Balaji Vidyapeeth, Puducherry. She received an ICMR grant in the worth of 1.5 crores, and also she earned ICMR DHR Long-Term International Fellowship for Young Biomedical Scientists recently, and UGCB SAP Research Fellowship for the Meritorious Scholar, and Summer Research Fellowship by IAS, and ICMR DHR Short-Term Fellowship, and a Fellow of American Society of Biochemistry and Molecular Biology. With the short notes, I request Dr. T.S. Anita to deliver a lecture.
Shall I start, Mubarak? Can I?
Yes, please.
Thank you so much, Mubarak, for a very short introduction. So shall I continue? I can go ahead, right?
Yeah, please share your screen.
Yeah, 1 second. Yeah, I'll just give a short introduction, maybe a welcome speech, and then maybe I'll share the screen.
Yeah, please.
Yeah, okay. So I'll. So fine.
Your presentation would be fine because.
Yeah, I'll just make it up. So a very good afternoon to all the esteemed organizers of this symposium, distinguished guests, and the participants who are present here. So before entering into the session, let me congratulate all the women people who are there. I wish you all a very happy International Women's Day. So myself, Anita, I'm very much honored to stand before you as a resource person for this National Symposium, and I extend my sincere thanks to a special mention always to Professor Hemalata, Dean, and Dr. Mubarak Ali, Assistant Professor from BSR Ahmed Crescent Institute of Technology, Chennai, for entrusting me with the privilege of addressing you on this special occasion. So today's symposium actually not only provides a platform for celebrating the remarkable contributions of women in science and technology, but it's also going to offer us an opportunity to explore the groundbreaking advancements in medical research. So the topic that I have chosen today for you for this symposium I'm going to discuss about is about drug repurposing, which is a promising frontier for treating cancer. Hope from the morning session you would have been listened about nanotechnology. So this is a bit away from the nanotechnology that I'm going to talk about, since it's going to be it's lunchtime. So I thought I'll make it a little more interesting. So throughout my presentation, I hope I share insights into the exciting development in this particular field of drug repurposing, and I'm going to highlight the impact of repositioning the existing drugs for the effective management of glioblastoma multiforme, which is a brain tumor. So before we go into, I would also like to once again express my gratitude to the organizers for their vision in convening this symposium. So recognizing and celebrating the achievements of women in science is not only commendable, but it's actually going to be very much essential for fostering diversity and excellence in our scientific endeavors. So now let's start our intellectual journey, and let's celebrate the spirit of collaboration, knowledge sharing, and immense possibilities. So thank you for this opportunity once again, and let's move on to the session now. Is it okay, Mubarak? Is it visible?
Yeah, absolutely fine.
Yeah, fine. Okay, the topic of today's session, what I'm going to talk is the evolution of drug repositioning, a promising frontier for treating glioblastoma. So what is this glioblastoma actually is that? Glioblastoma multiforme, it is a grade 4 astrocytoma. It arises from a multi-step tumorogenesis of glial cells from the brain, and it is considered to be the most lethal primary brain tumors in adults. If you see, the overall survival rate is only about 1 year after the diagnosis. So the current standard of care treatment available for glioblastoma multiforme is usually maximum surgical resection of the tuber mass, which will be followed by radio and chemotherapy with a particular single alkylating agent called as temozolomide. So if you see, so though there are vast treatment modalities available for cancer and particularly for GBM, if you see, the tumor recurrence is inevitable with a very poor prognosis of only 12 to 15 months. So because of its very poor prognosis, actually this put forth the need for the scientists to understand what the molecular complexity of this particular disease, and they wanted to design novel therapeutic strategies such that we are able to improve the overall survival of the patient with GBM. So because of that, if you see at the molecular level, GBM is characterized by aberrant activation of major signaling pathways and transduction pathways like PI3K, AKT-mTOR pathway, hedgehog pathway, Wnt signaling pathway, where all of which contributes to GBM proliferation and tumorogenesis. Since GBM is very difficult to treat, so developing of a multi-targeted therapeutic approaches, either using a combination of drugs or a known drug which has a broad spectrum of targets, will actually provide a solution to overcome the intrinsic and the acquired resistance for this particular tumor, that is GBM, to cytotoxic treatments. So while if you see, the search for alternative therapeutic strategies are still underway to suppress GBM pathogenesis, so previous studies have suggested that repurposing compounds of synthetic and natural origin actually would improve the therapeutic efficacy of the standard drug, that is temozolomide, and may also improve the overall prognosis in GB patients. So what is this drug repurposing or drug repositioning is called as? It's also called as polypharmacology, meaning it involves the use of previously approved drugs for new medical indications. So this repurposing drug strategy, if you take, it is very much advantageous because it is considered to be an alternate to de novo drug discovery. This is because we have already known the pharmacological profiles of the drug. We would know they are going to be less time intensive and they are going to be less costly also. So as a novel cutting edge therapy, so this technology actually holds a great promise in the future for treatment of tumors, many tumors, including GBM. And if you see, there are several clinical trials are underway with these repurposing drugs. So if you take the traditional drug discovery process, like how do the traditional drug discovery process takes is that, so first we go for a drug, it begins in the laboratory, and then we go on for the animal setup, and then we try to answer some basic questions. So that is going to be discovery and clinical, and it's going to be taking about average of about 6.5 years. So once after we have done the discovery and the preclinical study, and then we go on for the safety review, we check whether all the result, animal testing and all the other things are going to be safe. So once after that, then we move on to the phase 1 clinical trial where we try our drugs into the patients. So that will be with a very minimal number of patients, and the average should be around 1.5 years. So once we have tried and we are successful with the phase 1 clinical trial, then we move on to the phase 2 clinical trial. Again, we will be trying with a higher number of patients when compared to that of the phase 1. So once after that, that will be around average of 2 years. So once after that, the final one will be the phase 3 clinical trial. Again, it will be tested on the humans with a more number of population, and the average is going to be 3.5 years. So once we have tried with all the phase 1, phase 2, phase 3 clinical trial, then what happens? Now the FDA team enters here. So they examine all of our submitted data regarding the drug or whatever device that we are doing it, and they make a decision either to approve the drug or not to approve it. So once they have approved, then the FDA will enter here and they monitor all the drug and the device safety once the product are available for use by the public. So average, if you see, for a traditional drug to be discovered, it usually takes around 17 to 18 years for a drug to start from the laboratory and come to the market. So to minimize these effects, there comes our strategy for drug repurposing. So this drug repurposing, so if you see, the normal drug development process is very time consuming and it is very expensive, as I told in the previous slide. So on an average, it takes about 10 years and at least 1 billion to bring a drug to the market. This is, I'm talking about an average. So moreover, now if you see investment, pharmaceutical companies have started to invest more on drug development, especially in the field of drug repurposing, where they are able to find new uses for the existing drugs so that they can actually minimize their time limit of drug identification or drug development. And that process we call as drug repositioning or drug repurposing. So, and also due to the fast growth of bioinformatics and the biology big data, so this drug repurposing actually decreases the time cost of drug development process very significantly. And if you see now, researchers only need about 1 to 2 years to actually identify their drug targets and a minimum of going to be 8 years to develop a repositioned drug on an average. So this drug repurposing, actually, it is an untapped potential of working with what you know actually. So because we know the history of the drug in human, how it is going to work out, we know the safety profile and their side effects, and we also know that they're going to be very fast, cheap, they're going to be good for dry disease also. They are reduced the requirement for early stage clinical trial. In fact, repurposing drugs need not enter into phase 1 clinical trial also. They can directly enter into the phase 2. And we know the de novo drug discovery of how it has been discovered. And in some cases, we also know where their pathways of action actually takes place. So this is more advantageous for the researcher to go ahead with this particular drug repurposing strategy. So this actually shows some of the repurposed drugs. There are a few examples that I have listed out. So some common ones, like if you people know in the day-to-day life that we use, like if you take metformin, it is type 2 diabetes. Its original indication is type 2 diabetes that everybody takes very commonly now these days. So its new indication is it's been gone for PCOS, or sometimes recently it has been suggested for its anti-cancer activity also. And then there's another antibiotic, amphotericin B, which we use it for fungal infection. And now they've recently used it for leishmaniasis. So there are so many compounds that have been, so many drugs that have been already available. And now we are using it for a new purpose. So what is this drug repurposing, and what is the idea behind actually is that you take a known compound, you take a known drug like drug 1, drug 2, drug 3, you know their original indication, you make a combination of all three, and then you make it up for a new medical indications. This is actually the basic outline concept of drug repurposing. So coming into my topic, that is drug repurposing in GBM. So a drug has to be a drug which has to be an ideal candidate for glioblastoma must actually, should have the potency to cross the blood-brain barrier. So this blood-brain barrier is going to be a very thick membrane which doesn't allow all the drugs to enter. But an ideal drug candidate for particularly brain cancers, it has a potency to cross this blood-brain barrier, and it should accumulate appropriate accumulation in the brain such that its activity is going to be effective. And also it should not cause any significant toxicity also, because patients who are going to take up this clinical trial, they'll be willing to take only a drug when it doesn't cause any toxicity. Otherwise, why do they need to take those kind of drugs? So we need to choose a drug in such a way that it doesn't cause any toxicity to the patient. Hence, if you see, drug repurposing actually will present a compelling avenue for discovering effective therapies for glioblastoma. and it could very well lead to novel therapeutic paradigms for GBM. So the drug repurposing strategy, the big pharma way, and where I exactly stand is that so we have chosen some drugs from the literature. We have found out new target with old therapeutic indications, new target with new therapeutic indications, old target, new therapeutic indications. And then we have found out some evidences. We have collected all the evidences from the literature. And then we performed some in silico screening evidence based on whatever drugs we have chosen. And then we moved on in generating a proof of concept. So for that, we performed in vitro experimental studies. And from whatever data that we got, we then moved on to the preclinical experimental evidences. So from the preclinical experimental evidences, so from here, now we have actually started on moving with clinicians to go ahead for clinical trials. And if you see right now, currently, if you see, I'm at the preclinical experimental evidences where we have patented a particular combination of drug. And then there are I'm working with few consultancies where they want some more drugs to be tested. And I'm also serving as a CRO. So once this clinical trial works out, and definitely our combination of drugs is going to be a very good anti-tumor agent. And we are looking forward for that. So the drug currently that what we are working in my lab is that, so we work on metformin. You all know it's a first-line medication for type 2 diabetes. Chloroquine, it is an anti-malarial drug. You would have heard of it as well. Then is naringenin. It is a flavonoid actually present in citrus fruits like grapes, and it's a very good antioxidant. Another one is fluoroglucinol. This fluoroglucinol is also an antioxidant, and it is a phenolic derivative with antispasmodic activity, and it is derived from a marine compound. And then next one is epigallocatechin gallate. You would have heard of it. It is an antioxidant found in green and black tea. So my current research, this is just a thematic representation of my workflow. So what we exactly do is that we take up we take up glioma cells, and then we inject those glioma cells stereotactically, orthotopically into the rat's brain, and then we close the rat brain and then leave them for observation. So after 20 days of glioma implantation, then we treat the rats with a different combination of drugs. And then once the treatment period is over, we have targeted few cancer signaling pathways. So some of the drugs that I have used for my study is temozolomide. It is a standard drug, metformin, EGCG, chloroquine, naringenin, PGL, and fucoidan. So the research finding states that repurposed drugs targeting cancer signaling cascades, how we have actually dissected new mechanism of the repurposed drugs through in vitro and in vivo analysis. So first, initially, like I have just given 2 examples of whatever drugs that we are working on. This is all about my results and my research that I'm currently working on. So actually what we did is first, initially, we wanted to check how much of these drugs, these combination of drugs have a cytotoxic effect. So for that, we performed MTT analysis, and we found that the combination of our repurposed drugs, one combination is temozolomide, metformin, and EGCG. Another combination is a quadruple drug combination of temozolomide, chloroquine, naringenin, and PGL. So what we found was the combination of these repurposing drugs actually showed the best anti-proliferative effect at a lower dose when compared to the standard alone, as well as to the other treatment groups. I have not projected the other treatment groups here. So what I concluded from is that the combination of drugs actually exhibited potent anti-proliferative effect. So once we had determined the anti-proliferative effect, and then now we wanted to check whether our drug exhibited any apoptosis, whether it has induced any apoptosis in the glioma cells. So we had performed some apoptotic staining assays. We had done through fluorescence microscope and confocal microscopes. So there, interestingly, we found that our triple drug combination and a quadruple drug combination, they actually synergistically, they reduced the proliferation of the glioma cells, and they induced apoptosis. So as evident by a dual staining of acridine orange ETBR, Hext 33342, which was done through confocal microscopy. And then once we have known it has, now we confirm that, okay, fine, it has induced apoptosis. So now we wanted to check whether it is doing something with the mitochondria. So then we performed this JC1 analysis in a confocal microscope. And we also found that this triple drug combination and the quadruple combination of repurposed drugs, they actually promoted loss of mitochondrial membrane potential. So because of this membrane potential, loss of mitochondrial membrane potential, we wanted to check whether is it affecting the cell cycle arrest, or does it doesn't do anything with the cell cycle arrest. So we did some flow cytometry analysis, and then we found whether our drug actually is doing some kind of work with the cell cycle arrest in the glioma cells. So interestingly, we found that our repurposed drugs, combination of repurposed drugs, actually increase the percentage of cells in the G0 phase, increase the percentage of cells in the G0 phase. So this G0 phase actually leads to cell cycle arrest and apoptosis. So based on this, like, okay, fine, now we are sure that our drugs have actually, they have potent anti-proliferative effect and anti-apoptotic effect. So now, okay, fine, in vitro is done. So let's move on to the in vivo model. So we chose, we wanted to check what exactly is happening inside the rat model. So to study the molecular mechanism of drug action, so we took up in vivo GB models that actually mimic the patient tumor microenvironment. So we had actually established an orthotopic xenograft GB model using C6 glioma cells and treated them with the drugs. So how do we actually induce this xenograft induced model is that, so this is the C6 glioma cells of rat. So this is the stereotactic apparatus. We fix the animal, the rat, into the stereotactic apparatus at a particular bregma points in the brain. So once after that, we make a hole drilling into the rat brain. And then from the cell culture, we inject these C6 cells exactly at this particular point. We make a hole drilling with the help of a driller. And then we inject the C6 rat glioma cells into the rat brain. And then after that, we close back the rat skull and the brain. And then we leave it for post-operative care. So once after that, we observe the animals after tumor induction of about 20 days. And then after 20 days, we decapitate the animals, and then we remove the brain. And exactly, if you see, this is the site of tumor induction where we have actually made the drill exactly. So once after we have done the treatment strategies, so once after 20 days of this particular glioma implantation, we started giving the drugs for the particular treatment for a particular period of time based on the combination of drugs. So what we found was, like, from the in vitro analysis, we moved on to now into the preclinical model. So we have taken the rat brain tissues. So in the rat brain tissues, we had performed histopathological analysis and immunohistochemistry analysis for proliferation markers like Ki67 and angiogenesis marker, that is VEGF. And we found that the combinatorial treatment actually synergistically inhibited the glioma cell growth, glioma cell proliferation, and it has induced apoptosis in the GB bearing xenografts at the morphological level. So this is the IHC analysis where we performed for Ki67 and VEGF. So thus we conclude that the combination therapy decreased the cellular proliferation and angiogenesis in the tumor sections. So once, okay, fine, we have found at the morphological level, there's something happening with our drug as that of the in vitro. So now we wanted to choose where particularly exactly this drug of action is actually happening. So we have chosen 3 different pathways for 3 different combination of drugs. So one is PI3K AKT mTOR pathway in glioma. Another one is inflammatory pathway, and another one is Wnt beta-catenin signaling pathway. So first, coming to this PI3K mTOR pathway, actually this pathway has been emerged as one of the major regulatory pathways of the cell survival, growth, proliferation, and protein synthesis in both the normal and the GBM cells. So for this particular combination of drugs, for this particular cancer signaling pathway, we have chosen a triple drug combination of temozolomide, metformin, and EGCG. So actually, if you see in glioma cells, that is the brain tumors, the ligand goes and binds to the receptor. That is VEGF, vascular endothelial growth factor, goes and binds to the receptor. So it will phosphorylate and activate PI3K, and that will activate AKT. So this AKT molecule has an agonist called as PTIN. So this PTIN actually is being inhibited in a glioma cell. So then what happens is AKT will activate mTOR, and then mTOR will activate the tumor cell survival, meaning the proliferation continues. So this AKT actually inhibits the BAX and BAT, which are the anti-apoptotic proteins. So because of its inhibition, there is it doesn't induce apoptosis at all. So automatically the proliferation of the cancer cell continues. Whereas in case of a TME treated glioma cells, what we found was our TME has gone and bound to the VGEFR. We performed this with the help of an in silico analysis, and then we found that actually it phosphorylated and down-regulated PI3K, AKT, and interestingly, here we found was this TME actually activated the agonist, that is PTIN, where here in the glioma cells it was found to be inhibited, whereas in case of the TME treated, it was activated. So because of this activation, the tumor cell survival was not happening at all. And also the apoptosis induction was also happening because of this TME drug combination. So this we actually proved with the help of gene and protein expression analysis. So we found that this triple drug combination actually reduce the gene and protein levels of some of the key regulators of PI3K pathway. PI3K pathway like P10, PI3K, AKT, PDK1 and all, and enhance the levels of P10, which is an agonist of this pathway, as I said before, thereby inhibiting the PI3K pathway in a tumor-induced rat model. And also, if you see the triple drug combination, this PI3K also leads to apoptosis. So, as I said, the triple drug combination induced apoptosis by elevating the levels of pro-apoptotic markers and by reducing the levels of anti-apoptotic markers. So, this we had performed through real-time PCR, immunoblotting, and ELISA. So coming to the second signaling pathway that we worked on was Wnt or beta-catenin pathway in glioma. So I should thank ICMR at this juncture for giving me a great project for this to establish this model and the study. So for this, we had actually worked with the quadruple drug combination like temozolomide, chloroquine, naringenin, and fluorouracil. So this Wnt signaling pathway, if you take, it is of paramount importance in gliomogenesis as it enables the GB cells to recapitulate the embryonic process. It promotes glioma growth, invasion, tumor vasculature, and therapeutic resistance. So in glioma cells, if you see, the Wnt ligand goes and binds to the Frizzle receptors that is present in the plasma membrane. And because when the Wnt ligand goes and binds here, it activates beta-catenin. So this beta-catenin then it translocates into the nucleus, and then it will activate some of its target genes like c-MYC, cyclin D1, MMP97, CD44, and this actually leads to cell migration and addition. So, but interestingly, in case of our quadruple drug combination, what we found was our drug has gone and bound to this Wnt ligand Frizzle receptor, and it has actually inhibited beta-catenin such that it is not being translocated into the nucleus. So, because of that, the target genes are not being expressed. So, meaning here it has stopped proliferation. So this novel quadruple combination of temozolomide, chloroquine, naringenin, and fluoroglucinol, they reduce the GB proliferation in vitro and in a preclinical orthotropic GB xenograft-induced model. So this also we have confirmed through gene and the protein expression analysis. So the potential mechanism of the anti-glioma effect of the quadruple drug cocktail, the gene and protein expression of Wnt signaling markers were assessed. We found that the quadruple drug cocktail had significantly downregulated the expression levels of Wnt or beta-catenin genes like EGFR, beta-catenin, c-MYC, c-Jun, ERK1 and 2. So to streamline what is happening in the downstream of this Wnt signaling pathway, we performed, we wanted to check if the expression has some effect on apoptotic markers also. And we found that the quadruple drug cocktail had induced apoptosis by caspase cascade and the suppression of anti-apoptotic protein BCL2. So now we have found like our drugs, okay, fine, they are able to, at the gene and the protein level, they're able to inhibit the signaling pathway. So now we wanted to really check whether the drugs have actually crossed the blood-brain barrier or how much do they actually cross the blood-brain barrier. So we had performed this ESIQ-TOF-MS analysis where we found that the systemic delivery of the drug as a combinatorial treatment, they were actually able to reach the target site, that is the blood-brain barrier. And they have, since they have crossed the blood-brain barrier, they were able to elicit the therapeutic responses. So actually a drug cannot as such directly enter into the blood-brain barrier. It has to enter through its metabolites. So we had chosen some of their metabolites like TMZ metabolite is AIC, CHL metabolite is D-acetyl CHL. So there are different metabolites. So based on the metabolite concentration only, we were able to identify, okay, fine, that drug has actually crossed the blood-brain barrier. So fine, after identification of the drug-brain barrier, then we wanted to check, okay, how far is a drug actually making the animal to survive? Because that is the ultimate point for a drug to come to the market. So we checked for a survival analysis and this survival analysis was plotted using a survival graph with the help of Kaplan-Meier curve. So here actually, this point actually indicates the death of a rat in each treatment group was considered as a breakpoint. So we had actually found that our triple drug combination and the quadruple drug combination, actually they were bestowed with the potential to provide superior survival to the GB xenografts. And this is all about the in vitro and the in vivo. Okay, fine, we have found survival statistics. So what is exactly happening at the membrane level, what is exactly happening at the outside of the cell, we wanted to know. So we had performed some molecular docking analysis of human VGFR receptor. So what we did was the in silico analysis, like based on the glide score, we found that the triple drug combination have evolved as a potential human VGFR inhibitor cocktail for GB patients in future. So while a docking study revealed that all the drugs with triple drug combination, they were able to significantly inhibit the glioma proliferation because it was able to bind particularly to the human VGFR protein, which is a key regulator of the PI3K pathway. So, so this is what I'm currently working on. So my concluding remark for this repurposing strategy, which is going to be the future now. So for GBM, as I said before, it is a deadly primary parenchymal CNS neoplasm with a very dismal prognosis. So by bypassing the time-consuming chemical optimization and toxicology testing in drug development steps, repurposing of existing approved drugs can actually help us to better manage any tumors, including GBM. So as compounds need to be brain penetrant to have maximum efficiency in GBM, so it is ideal to select a compound that are able to cross the blood-brain barrier and are not substrates of any efflux transporters. So furthermore, identifying pathways controlled by each drug is very much essential for identifying some therapeutic combinations for treating GBM. So from my study, what we concluded was that the combination of drugs, they were actually able to inactivate the PI3K pathway or Venn signaling pathway, paving a way for apoptosis at the preclinical orthotropic GB xenograph model. So as a proof of concept, repurposing compounds of synthetic and natural origin as a cocktail will actually synergistically will enhance the anti-GB potency of the standard drug, both in vitro and in vivo. So actually, as a future perspective, we are taking out these drugs into clinical trials. We are in a connection talking with the doctors for the clinical trial and trying to get the approval for the clinical trial. And in summary, if you see, repurposing drug is going to be the future. So drug repurposing that opened a window for drug discovery is a trend for including, but not limited to cancer therapy, regardless of many failures happening so far. So as Nobel laureate in physiology and medicine, James Black once said, the most fruitful basis for the discovery of a new drug is to start with an old drug. So I totally concur with this concept, and with that concept only I started working on in my laboratory, in my research. So so some of the animal models that we are currently working on in my laboratory is one is xenograft-induced animal model for glioma, and currently we are working with orthotropic xenograft-induced HCC hepatocellular carcinoma. Now we are actually shifted on to patient-derived xenograft models also. And then we are also working on selenite-induced cataract model. So people who want to work with animal and want to collaborate with in our laboratory, you're most welcome. And some of my references and my acknowledgement, I should actually thank my PhD students in a very much early career. I should thank my PhD students like Dr. Shreyas Kiskodwali, Dr. Daisy Priscilla, and Ms. Indrani Bisos, who was also completed. So I should thank these three people. Without these three people, I couldn't have done so much of work in my early career. And I thank the other colleagues of my previous institution where I started working all these stuffs, and also my present institution, that is Pondicherry University. And I would like to thank ICMR for giving me a grant, and Dr. Vani Adityan Research Grant from SPV, Puducherry. And the greatest, my greatest position is always going to be my family, and I thank them. So some of the life examples of repurposing, and thank you.
Thank you so much, Dr. Anita, for your super fast presentation.
Oh, it is so fast. You could have interrupted me actually, and you could have told.
No, it's great, and it's a new thing that among all other presentations, maybe
Okay.
the participant might enjoy your
Hope it has done too.
Yeah,
I've justified.
Absolutely. So, the topic is now open for the discussion. If you have any questions, yeah, just they're thanking you. Maybe they're willing to go for lunch.
Yeah.
Yeah, because it's thank you for the scheduled completion of your talk as well.
Yeah. No, yeah, I know it's like, lunchtime. I shouldn't force people to sit right in my presentation.
Yeah, fine. Okay. Maybe when participant asks question, I will post to you.
Yeah, definitely.
Maybe
Definitely.
you can allow them to go for lunch.
Yeah.
Thank you so much again for your presentation.
Thank you, Mubarak. Thank you so much for this opportunity. And I would like to thank Hemlata ma'am also for this opportunity. And once again, wish you all a very happy Women's Day. Have a great day.
Thank you so much.
Thank you, Mubarak. Yeah, thank you.
Yeah, the note to the participants, actually now this presentation our session is going for lunch, and then you'll be asked to assemble the same window at 2:20. So the lunch time is very huge. Now we can join at 2:20. So by the way, the one more talk by Dr. Radharani from NIT. As soon as the presentation over, we're going to have concluding remarks by our dean. Then there will be a top 5 slogan will be displayed, and then will be acknowledged in the valedictory. After that, we provide you the feedback link. Those who are filing the feedback, they're gonna get their certificates to their email. So I request you to come assemble at 2:20 to the same window. Thank you so much. Hello.
Hello, am I
Yes.
audible?
Yeah. Yes, ma'am. Yes.
Okay.
Okay. Thank you. The students are joining after the lunch.
Okay.
So anyhow, we can start our event because Dr. Radhani have other commitments to complete it. Anyhow, let's welcome Dr. Radharani from Associate Professor, Department of Biotechnology, Motilal Nehru National Institute of Technology, Allahabad, Uttar Pradesh. Actually, the area of interest is about environmental biotechnology, bioremediation of biological wastewater treatment, and specifically microwave fuel cells and the plant microbe interactions. So she have completed her PG from Nagpur University, where she got more gold medalist, and PhD in microbiology at CSIR NEERI, that is National Environmental Engineering and Research Institutes, Nagpur. And she have received grants from various funding agencies, which include TechCube, TAE, DST, DBT, UTST, and Ganga Mission also, the worth of 2.1 crore. And she has the awardee of CSR SRF and a travel grant from DST to visit Germany. And also she sanctioned a project with the SERP DST Yearly Career Research Award. With this, I'd like to invite Dr. Radharani for giving her lecture.
Thank you very much, Dr. Mubarak Ali, for such a wonderful introduction. A very good afternoon to all, and wishing you all a very happy Women's Day. So, first of all, I would like to take this opportunity to congratulate the organizers of this e-Symposium on Women Scientists in Science and Technology, organized by B.S. Abdur Rahman Institute of Science and Technology, Nano and Biomaterials Association, and BRSI, that is Biotech Research Society of India. Particularly, I would like to thank the convener of the symposium, Professor S. Hemlata, and coordinator and organizer, Dr. Mubarak Ali, for having me as invited speaker for today's event. So, as Dr. Mubarak Ali has already introduced me, I'm Dr. Radharani, and I work as Associate Professor at Department of Biotechnology at MNNIT Allahabad, Prayagraj, Uttar Pradesh. And a broad area of my research is environmental biotechnology, with particular focus on bioremediation and phytoremediation, biological wastewater treatment, bioelectrochemical systems, and plant microbe interactions. And in today's, let me share my screen first. Yeah. Is my screen visible?
Yes.
Okay. So today I'm going to talk about microbial fuel cells. As I told you that I'm also working on bioelectrochemical systems, particularly microbial fuel cells. And as we have seen, we have listened to many speakers, including Professor Rukmani, Dr. Jayashree Raut, and T. Anita, and all of them have given a good background about nanomaterials and their widespread application. These days, biomaterials, specifically nanocomposites, are finding application in each and every aspect of life, whether it may be the medical, diagnostics, therapeutics, enzymes, catalysis, agriculture, or anything. Nanomaterials play a very important role in it. And I'm not a person of nanotechnology in particular, but as these materials are very versatile, they can find application in any aspect of life. So we have also done some work on application of nanocomposites for increasing the reactor kinetics, or particularly electrode kinetics, in a microbial fuel cell. So I'll be talking about microbial fuel cells and their applications in general, and giving some glimpses of the work which we have done on electrode modification using nanocomposites. So for today's talk, the topic is microbial fuel cell advances in electrode modification using nanocomposites. The slide is not moving.
We can scroll down, it will be changes.
Can someone suggest? Yeah, now it has started moving. Sometimes there are certain technical glitches in online sessions. Yeah. So this slide shows an overview of the global energy utilization and energy demand and the projected energy demand. So in today's scenario, the current scenario, we are highly dependent on largely 85% of the energy is derived from conventional and conventional sources like fossil fuels, including coal, natural gas, and oil, and a small fraction is derived from the renewable energy sources. And as a projection, we see that in upcoming decades, we are going to see a drastic increase in energy utilization and energy demand. So we need to be prepared with more research and more options so that we can lower down the burden on our fossil fuels or the conventional non-renewable energy sources. And we need to increase our dependence on the renewable energy sources. And among these renewable energy sources, we know we have very much well-established technologies like the wind, water, hydel, that is known, geothermal, and nuclear. But an emerging technology is that of microbial fuel cells. Now, what is a microbial fuel cell? Microbial fuel cell is a bioelectrochemical device that converts chemical energy present in organic compounds into electrical energy by microbial metabolism. So basically the electrons which are produced, they are because of the microbial metabolism. There are certain set of microbes commonly known as exoelectrogens which have this capability of producing extracellular electrons during the metabolic processes. And these electrons can be harvested, they can be allowed to move through an external circuit, thus leading to the flow of electrons and hence the flow of electricity. So this is a general overview or a schematic representation of a simplest model of microbial fuel cell. Here we see we have two compartments, the anodic compartment and the cathodic compartment. The electrode in the anodic compartment is anode, while the electrode in the cathodic compartment is cathode. In anode, the microbial activity happens. So all these exoelectrogenic microbes, they are present in anode, and the extracellular electrons, they are produced only under anaerobic metabolism. So that is why an anode has to be, or anodic chamber has to be anaerobic, while the cathodic chamber has to be aerobic because oxygen acts as a good electron capturing species and it can capture electrons. So these exoelectrogens, they act as biocatalyst, they produce electrons. Electrons are allowed to flow through an external circuit and they come to cathode where they are captured by oxygen or other electron capturing species. Oxygen, if present, it is reduced to water. While during metabolism, along with electrons, protons are also present and they are allowed to pass from the anodic chamber to the cathodic chamber through a proton exchange membrane. So this is a general overview of how a microbial fuel cell functions or works. So majority of the things I have already discussed. As we can see in the anodic chamber, the reaction follows like this. Here we have taken an example of a simplest carbon and energy source for microorganisms, that is glucose. It leads to production of electrons along with protons under anaerobic conditions, whereas in cathode these electrons combine with protons to form water. Now coming to the materials, the anode and cathode material, that is the electrode material, it can be made up of, largely it is made up of carbon materials. So the carbon can be in the form of carbon cloth, carbon brush, mesh, felt, paper, or graphite and its derivatives, graphene, and sometimes metals, conductive metals are also used as electrode material. Then for ion exchange membrane, the common things which are used include Nafion, Ultrex membrane, Nafion membrane, and salt bridge. The body can be made up of any neutral material, including glass, plexiglass, plastic, acrylic sheet, or ceramic. So I have already described this. In anode compartment, the bacteria utilizes organic substrate and produces electron. These electrons are transferred to the anode. Then from anode, the electrons go to the cathode, while the protons travel via the proton exchange membrane. Now, yet another factor which is important in a functioning of a microbial fuel cell is the transfer of electrons from microbes to anode. This electron transfer has to be efficient so as the system can function properly and give a proper or a good power output. So this electron transfer, it can be by two mechanisms: the direct electron transfer mechanism or the indirect one, which is mediated electron transfer mechanism. Certain group of microorganisms, they have direct means by which they directly adhere or attach to the anodic surface and perform direct transfer of electrons. Like there can be elongated pili-like structures, which are often known as nanowires, which directly transfer electron from the microbe to the anode. Then certain electron carriers of bacterial origin, which can be present on the outer surface, like cytochromes, also play an important role in this. While for mediated electron transfers, like in many microorganisms, direct transfer is not possible. So we need to add mediators, certain organic compounds which can exist in both reduced and oxidized states, so that they can carry electron from the microbe to the anode. So that way we need to modulate, or we need to design the system so as the electron transport from the microbe to the anode is efficient. Now, with this background, now we understand that microbial fuel cell is an emerging technology and it has several advantages and applications. The very first thing is it is an environment friendly and cost effective technology which can be sustainably used to produce green electricity with minimum requirement. Like as anolite, we can use wastewater, which can be either of agriculture origin, the domestic sewage origin, or industrial origin. So in that way, we can convert, we can get good energy recovery and low operation cost, because the overall process is sustainable because of the production of energy. Then green electricity is produced, the carbon footprint is low. So we see the environmental impacts are good. It is a eco-friendly technology. The economics are also fine. Energy output is good. And also it has potential for the treatment of wastewater. And MFCs can also be used as biosensors for sensing pollutants in wastewater. So the technology has several applications as well as several advantages, but at the same time, it has certain limitations and challenges which limit its application at field scale or a large scale. And the most important limitations include the high internal resistance and low power generation. So this is the major hindrance in application of the technology as field level, that because of the high internal resistance, power generation or the power density which is obtained is relatively low. Then another issue which is, or another challenge which is often associated with MFCs is the biofouling of electrode surfaces during extended use or during prolonged applications. And obviously the design cost of, or the initial investment cost of installing a MFC system at a large scale in field. So these all are the challenges which are faced by researchers and workers when we want to implement this technology to the field. And people are working on various aspects. Some people, they are working on process parameter optimization. Then people are working on development of efficient electrode material or electrode catalyst to improve the catalytic performance and get a good yield by minimizing internal resistance. And very much work has been done, quite much work has been done in reference to modification of the electrode material. There can be various approaches, but one of an important approach is modification of electrode kinetics. So cathode and anode has been modified in various ways, and one of the common modification which is very much into research these days, and we have also done some work on it, it is by using nanocomposites or nanomaterials to improve the electrocatalytic property of the system or to improve the electrode kinetics. So the electrode material and electrode modification, these are two important parameters which are going to significantly affect the performance of the reactor. Apart from this, other important things are the separator or the proton exchange membrane. It also has important role in increasing the power density output. And yet another factor is the use of high efficiency exoelectrogenic strains. But here we are going to focus more on cathode and anode modification using nanocomposites or nanomaterials. So currently there are different materials which are used as electrodes, both anode and cathode, in a microbial fuel cell, like carbon paper, carbon cloth, various forms of carbon, carbon fiber, reticulated vitrified carbon, carbon mesh, then graphite in various forms, like rod, carbon brush, crystalline or polycrystalline graphite. Metal electrodes are also common, so zinc, nickel, copper, silver, iron, aluminium, stainless steel. These are common metal or metallic electrodes which are basically used. And conductive polymer based strips are also very much used in microbial fuel cell as electrode material. So what exactly should be the properties of a material which so that it makes it of utility to be used as an electrode? So it should be highly durable. Obviously, it should be non-toxic in biocompatibility. So as far as the non-toxicity or biocompatibility is concerned, particularly the biocompatibility, it is important from a perspective of anode, because anode is being dipped in or being immersed in a solution where microorganisms are cultivated or allowed to grow. So biocompatibility is essential and cannot be compromised as far as the anode is concerned, because anodic electrode or the anode should be a base, or it forms a base for forming a biofilm of exoelectrogenic microbes. So this property is of extreme relevance for anode. Corrosion resistance, again required for both anode and cathode, because as we know, a biofilm is formed on anode, so it is very much prone to or very much susceptible to degradation or deterioration if used for a long period of time. So corrosion resistance and high durability are essential for long-term and field use or field application. High conductivity and increased surface area is also important. Increased surface area is required for efficient electrode reaction to happen, especially in anode where the microbes are going to give electrons or attach themselves. Then it should not be toxic. That means it should be environmentally and ecologically friendly. It should be low cost. So whenever we are going for a large scale application or a field scale application of any technology, the material which is to be used should be economically viable. and improved electron transfer, so that internal resistance is decreased and efficient power output are obtained. So with this background information about microbial fuel cells, now we are going to focus on how research is going on and how nanocomposites have proven to be an essential and important component in enhancing electrode characteristics. Largely, nanocomposites, as we know, they have wide kind of catalytic activity, and they have improved conductivity, improved electrocatalytic activity, and high surface area. And this very property of nanocomposites, that is high surface area to volume ratio, makes them apt candidate to be used for electrode modification. Then biocompatibility support is also achieved by use of certain nanomaterials, certain modified nanomaterials, which is important for enhanced bacterial attachment and growth, which is required for the development of a biofilm over anodic surface. Then durability and stability, which includes enhanced mechanical strength, enhanced chemical stability, and increased resistance to fouling and degradation is also a desirable property. Then enhanced mass transport, which is achieved because of the tailored porosity and permeability of nanocomposites, which can facilitate the transport of nutrients, ions, and electrons within a MFC system. So all these things, all these properties, they can be imparted by the use of desired nanocomposites or engineered nanosystems. So largely, as far as the electrocatalytic properties are concerned, we have seen that metal oxides are of extreme importance, and various metal oxides have been used as electrode modifiers. But apart from metal oxides and graphene and modified forms of graphene, there are several conductive polymers as well, or polymer-based materials, which also are widely used and have been well researched, particularly polyaniline, polypyrrole, and polythiophene. So here we see a small or a brief timeline depicting the development of electrode material. So this is the timeline for anode, and this is the timeline for cathode. And we have a look, if we have a look at this timeline, we see that largely the carbon-based materials, which are acting as the base layer, that is the carbon cloth, carbon felt, here again, carbon cloth, they are modified using either metals and largely metal oxide like manganese oxide. Here, polyalanine is used in combination with carbon cloth. So here we see in this timeline that largely the research is focused on the use of either conductive polymers or metal oxides, or combination of conductive polymers and metal oxides with graphene or modified forms of graphene. Extensive work has been done on these types of nanostructures, and still people are still working, and more studies are getting published every day. Whereas in case of cathode, where largely the modification is done to improve the oxidation reduction potential. Here, metal oxides are of extreme importance. And one of the main focus for cathode modification is to decrease its cost, because most of the times the metal which is associated with cathode are very costly materials like silver and platinum. These days also the platinum, though in very small quantities, but it is used because it provides a very good OR potential, and it removes electrons very efficiently, and hence the electron flow is very efficient. So for cathode, the improved OR potential is to be targeted, and for anode, improved biocompatibility, efficient electron transfer, and high porosity and high surface area is the target, which needs to be fulfilled or achieved. So now I'll be more focusing on what work we have done in our lab with respect to the modification of electrodes in a microbial fuel cell and what kind of observations we have achieved. So this is a paper published in Bioresource Technology about optimization of operating conditions of miniaturized single chamber microbial fuel cell, where nickel tungsten oxide in combination with graphene oxide was used as a modifier for carbon cloth electrode, and it was found to improve the microbial cell performance as well as it improved the microbial community dynamics within the microbial fuel cell. So on the basis of the electrochemical findings, so electrochemical findings here were performed in an electrochemical analyzer, and here we performed largely the cyclic voltammetry and LSV studied. And it was found that when the modified electrode was used as anode, the power density was found to increase by 8.5 folds in comparison to the control. And this significant improvement which occurred in the electron transfer between the microbe and the modified anode, it was because of decline or decrease in the internal resistance, which was evident or validated by electrochemical studies. So here the experiment was performed in a very miniaturized system. In fact, this microbial fuel cell system of ours was so small, it was a very small scale system. The total volume of the system was 0.8 ml, and we got good results in that. So showing that NWO4 and graphene oxide have good potential, they have good electrocatalytic property, and it has good potential to be used in MFC or similar cells, similar fuel cells to be more specific. And here we can see some of the TEM images of modified electrodes. Here we see the TEM image of reduced graphene oxide, and here reduced graphene oxide with NWG, that is nickel tungsten oxide, the combination of reduced graphene oxide with nickel tungsten oxide. These are the various concentrations of the metal oxide, and hence we can see as the concentration of metal oxide is increasing, the dot layer or the points on the graphene sheet, they are seen to increase. And this is the SAED pattern. So here the TEM images of different equimolar concentrations of nickel and tungsten oxides on geo sheets as visible, they clearly show that the metal oxide has been uniformly and evenly deposited on the graphene oxide sheet, which led to the improved surface area and improved electrocatalytic property, which was observed as a part of our findings. In this study, which is published in Journal of Environmental Chemical Engineering, we have used the same composite, that is nickel tungsten oxide with reduced graphene oxide nanocomposites as electrocatalyst. And we have seen the effect of this particular nanocomposite as an anode modifier on the population, the microbial population present in the anolyte. And obviously the current was high, and as I have told earlier, that high increase in the current density or the power density which was observed, it was well validated by electrochemical studies and electrochemical data, but it was also found on the basis of the NGS studies which was performed when the whole anolyte was taken, the biofilm from the anode was taken, and it was subjected to microbial community study. It was found that the anode catalyst increased the relative abundance of gamma proteobacteria. And initially, the population of gamma proteobacteria was not so high, but as the system was allowed to operate over a period of time, the population shift was observed and population was found to shift towards gamma proteobacteria. And we know that gamma proteobacteria is abundant in many genera of exoelectrogenic microbes. So this shows that sometimes some kind of electrochemical modification can also lead to change or shift in the microbial community dynamics, thereby increasing the number and diversity of desired group of microbes, that is exoelectrogenic microbes. In yet another study performed by our group, which was published in International Journal of Hydrogen Energy, another type of modification was performed or was studied. It was a conjugation of molybdenum oxide with polyaniline. So here we have used or we have tried the conductive polymer as well. And here also we have seen a good electrocatalytic potential as well as good biocompatibility potential of this combination of nanocomposite, and it was found to increase the power density significantly in the modified electrode in comparison with the native electrode or the bare electrode. And the electrochemical studies showed that the charge transfer resistance of the modified single chamber microbial fuel cell was significantly reduced. So the charge transfer resistance was drastically reduced. And as we have seen that internal resistance is one of the major limiting factors which declines the efficiency of any microbial fuel system. So we need to get rid of this limitation. We need to somehow reduce the internal resistance, and it can be done by using modifiers or using nanomaterials and nanocomposites as electrode modifiers. Here, let us have a look at the SEM images, FE-SEM images of these nanocomposites. So here we have molybdenum oxide and polyalanine in different magnifications. So these are the FE-SEM images. So these figures show very well that these rod shaped, these are the rods of molybdenum oxide, so that polyalanine gets coated over the molybdenum oxide rod structure, as it is quite much visible here in this image, that is image C. And this gives rise to a scaffold-like structure, which creates a large conductive surface area, which is desirable for functioning of anode, or for that matter, efficient functioning of anode. This paper is also a part of initial study only, that is the electrode modification or the anode modification, which was done using nickel tungsten oxide reduced graphene oxide coated carbon cloth anode. And here we can see in the SEM images, it is very clear that how on carbon cloth, the modification of carbon cloth leads to increase in biocompatibility, which is evident by the biofilm or the microbes which are adhered or adsorbed on the surface of the modified electrode. So though in all these studies or in all these papers, data is presented with respect to anode modification, but in our lab, we are also working on cathode modification, and our main target in our studies for cathode modification is to reduce the dependence on very costly elements. As I told you, that platinum is very commonly used, or platinized carbon is used as a catalyst in cathode. So we are trying to use other alternatives, um, other metal oxides or other metals, and somehow trying to get the same amount of efficiencies in terms of OR potential as far as cathode is concerned. But I have not included the results because the studies are under process and we are yet to receive the conclusive findings. In this study, which is published in Journal of Cleaner Production, with the title Electrochemical Behavior of a Multimodular Microbial Fuel Cell Operated Using Cow Dung Enriched Inoculum, Enhanced Energy Recovery Using Electrochemical Optimization. So here we have focused more on the biocatalyst. As initially we were discussing about the pros and cons or the advantages and limitations of microbial fuel cell, we discussed that among limitations, one of the important limitation is internal resistance, which can be combated by using advanced materials. But one another important thing which is significant as far as the whole system is functional is the microorganisms, because here microorganisms, they act as biocatalyst. So here we have talked about, or we have discussed the operation of a multimodular microbial fuel cell using a population of exoelectrogenic microbes, which we have enriched from cow dung. And we have found good results. So a liter scale multimodular microbial fuel cell with cow dung inoculum was operated, and it was worked out under different circuit connections, and detailed electrochemical analysis was performed. And we have reported a very good power density output in this study using a simple carbon cloth electrode, and modifications can obviously lead to further good findings as far as the energy output is concerned, and also good COD removal. So 60%, 67% of COD removal was observed. So whenever we talk of a MFC and we say that it can be used for generation of electricity, at the same time it can also be used for the removal of pollutants. These two things, they go conversely. If from a system we are getting good amount of electricity, then obviously it's not going to lead to a significant amount of COD removal, and vice versa. So researchers are also trying to optimize the system so that maximum benefit can be extracted out of it. And here we have reported relatively a good power density and an appreciable amount of COD removal. Those who are interested in this topic and want to do research on improvement or development of electrode materials, in this review paper of our group, we have reviewed about different nanomaterials and different kind of other materials which have been used as modifiers for electrode for improving the reactor kinetics in microbial fuel cell. It is published in Chemosphere. So as we have discussed, that anode modification is largely aimed for efficient electron transfer by the catalyst, the biocatalyst, which is the microorganism. And modification of cathode leads to increased oxidation reduction reaction rate. And largely the kind of materials which have been efficiently used and given good results include conductive polymers, carbon, and metal-based materials or metal-based nanocomposites. Now, coming to the concluding remarks, I would like to emphasize that in this presentation we have seen and I have described about the importance of MFC for economic and eco-friendly power generation along with wastewater treatment and resource recovery. Further research is required in this field to develop and optimize the existing systems so that they can be implemented at field scale. And the most important thing which makes the system functional are the exoelectrogenic microbes, also known as biocatalyst. And to name a few, because I have not included name of exoelectrogens, so I would like to mention here in the concluding remark, the very widely studied and commonly studied exoelectrogenic microbes include Shivanella, certain species of Pseudomonas and Geobacter. Extensive work has been done on these group of bacteria, and they play a very important role in power generation, and hence study of these exoelectrogens is also important. Then for optimizing electrode material, changing and designing the process parameter is also important to get efficient output. And here in modifying the electrodes, in modification of electrodes, nanotechnology holds immense potential, because by using an array of nanocomposites having various kind of properties in terms of electrocatalytic efficiency, in terms of biocompatibility, in terms of high surface area, there is immense potential. And nanotechnology and nanocomposites, of course, can be used to improve the performance of MFC. And microbial fuel cell is an emerging technology, and it is a very promising tool to cater the needs of sustainability. And it is also important to come up with solutions to harness the maximum potential of the same, which is yet underutilized and yet waiting to come on the field. Now coming to acknowledgements, the work which I have shown was funded by two of the SERB funded projects. The grant number is given here. And I'm also grateful to my host institution, that is MNIT Allahabad, for providing infrastructural facilities and all other kind of support. And whatever data I have shown and presented, or whatever papers I have presented here or shown here in this presentation, are from the PhD work of two of my PhD students, Dr. Gitanjali and Dr. Roma. These are the references. And with this, I conclude my talk. Thank you very much for your patient listening, and I'll be happy to take a few questions.
Yeah, thank you so much, Dr. Adarani, for excellent presentation. Now the topic is open for the discussion. The participants can raise your questions. Even you can post your question in the chat box. So meanwhile, the type, I have some questions. Like,
yes, sir.
Actually, based on you're changing the nanocomposites, you are talking about only power density. So with that only, you can able to say that this much power is generated by applying these composites.
See, nanocomposites can be of different types, and they can provide different kind of modification. So we need to target what kind of property we would like to improve or change. Like in anode, we are looking for the electron storage and electron capture potential. If a particular nanocomposite is biocompatible, at the same time it is very much efficient in its electrocatalytic potential, it will very evidently and very strongly extract electron from the microbes, and hence improve the functioning of a MFC. Similarly, if you want to modify the cathode, we are looking for a good OR rate, oxidation reduction potential. so that when the electron which is traveling from anode to cathode is very strongly pulled. So here, if the pull from the cathode is high, then the flow of electrons will be uniform. And if flow of electrons is good, it will lead to a stable energy or electricity generation.
Yeah, but the nickel, I mean, because of 67% of the PD increment in your study, it because of that composites or any other reasons?
According to our findings or whatever studies we have performed, it is because of the reduced internal resistance. So the electron flow was facilitated. When internal resistance is reduced, the flow of electrons is facilitated, which leads to improved output.
Yeah. But most of your composites are with the nickel tungsten
Yes,
oxides.
largely
But
that work was on nickel.
yeah, because of the magnetic property to increase the increment of that PD, or
No, here we are not. We have not studied about the magnetic properties. Largely, we have studied about the electric properties and the capacitive
no,
properties.
nickel itself they have magnetic property, right?
Okay.
So because of that might be the increment of PD
Yeah,
maybe
that can be the reason, and that is the reason why nickel
literature.
composites are extensively used in fuel cells and capacitors. It can be one of the reasons.
And what is the challenges when using the graphite, reduced graphite than any other composites?
Well, people are using CNTs,
Yeah,
and
yeah.
reduced graphene oxide is also very commonly
Mm.
employed. But whenever we talk of large scale applications, we must be bothered about, or we must be concerned about the cost effectivity and as well as environmental compatibility. So and we all know that nano composites, this is the limitation of nano composites, that for many of these composites the large scale toxicological assessments have not been performed. So this is a big limitation, and it is going to be a deciding factor whenever we are going to use any technology in large scale. At experimental scale, at lab scale, we can go for any of the modification and report, okay, we are getting good results with this particular nano composite.
Yeah. Yes. Okay, thank you, ma'am. Maybe the participants are only given appreciation. They don't have any question. Maybe they will raise you.
Sure, I am anytime available for your queries. You can drop me a mail or anything.
Yeah.
Okay, thank you so much for having me.
Yes. Thank you, ma'am. Actually, we're going to close this session. Maybe you can find time. You can spend few more.
Sure. I am here only.
Yeah, you can. Maybe you can stop share your slides.
Yeah. Hmm.
Ma'am, are you available, ma'am? Sorry, the lectures are over. We had to have some concluding remarks, and also we're gonna share the best slogan presenters, and we share the feedback link for the certificates. Please be there for 5 more minutes. Thank you. We're very sorry for the delay. Let's proceed for the slogan, best top 5 slogans. Actually, we have received a 384 registration, and among which we got 250 slogans, and many of them not interested to write it. But however, 250 plus slogans, we have derived 5 as the best. So the one is the Empower Her Today, Tomorrow, Together, written by Mohammad Yunus Jafri from Bansal Institute of Engineering, Lucknow. And No Woman, No Existence from Gaurav Verma from EKTU, Kanpur, UP. From Struggle to Strength, Honoring Women's Achievements. Meenakshwari Konjanba from lovely Professional University, Punjab. Empowered Women of Today Have an Absolute Sense of Mission by Anil Kumar Sharma from Amit University, Punjab. Women are not only the mother but also a leader by Aishi Singh from Bansal Institute of Engineering, Lucknow. So it was adjudged by Dr. M.K. Sankida. So I thank for her help. And actually, we're going to have this concluded remarks, but due to some administrative hectics, she could not make it herself. But I like to thank all the speakers, right from Professor Rukmani ma'am, elaborated the drug delivery systems, and Professor Jayasri Rao for biosynthesis of nanomaterial using algae and lichenology, lichens. And thirdly, Dr. T.S. Anita from Pondicherry University should talked about glioblastomas and the treatment using therapeutic drugs. And finally, we had a talk on MFC, that is microbial fuel cells using biocomposites, and enhances efficiency by Dr. Radharani. So I wholeheartedly thank all these speakers who immediately accepted my request to have, because since they have their own commitments today, even they have a holiday. So she committed to come over here, join here and deliver a talk on her interest. And lastly and most importantly, the participants. So throughout the continuous talk, they will be sitting and listening to the speakers, and also the students have joined in YouTube live. So for everyone, I am thanking for your wonderful participation in this precious occasion. So you can have this feedback. So once you completed your feedback within 5 working days, you're able to get your certificates, e-certificate you can receive in your email. So please enter your email ID very sensibly, because it's the only connecting point where you can get the certificates, okay, and also names. So thank you so much. I also post this link in the chat box. Thank you. I hope you are getting feedback link. Please note it.
It says that it no longer is accepting the feedback link, sir.
Sorry, ma'am. Now we can check, ma'am. Please.
Okay, thank you, Dr. Mubarak Ali.
Yes, ma'am. So that the link now accepting the responses, please do try now.